main topic for the day, which is the review of experimental evidence scoring for the ClinGen gene disease clinical validity framework. So as I mentioned, a few of us have put together some examples that we will go through and discuss, and would welcome any discussion or questions as we go through this. So the first thing I wanted to show is this table, which of course is in the gene curation SOP, just as a reminder of the different categories under which we collect experimental evidence, with this experimental evidence supporting the role of the gene in the disease. So broadly, the categories that we collect evidence under are function, which includes biochemical function, protein interaction and expression, and then functional alteration in patient cells and non-patient cells. We also collect information on models, including non-human animal model organisms or a cell culture model, and then rescue, which can be a rescue in human, non-human model, cell culture, or patient cells. So as I mentioned, what we want to do today is to walk through each category of experimental evidence to give some examples. But please do stop us if you have any questions or if you have any examples that you wanted to share. So before I go ahead and start with the examples, I did want to show this screenshot from the gene curation interface. I know that many of you are already using the GCI and are familiar with the interface, but just as a reminder, when you go into the GCI, you can choose the appropriate category for entering your experimental evidence and scoring it. And then the GCI is really helpful in that it really walks you through the different types of information that you need to give by just giving you prompts along the way. So it's pretty straightforward. And of course, if you ever have any questions about using the GCI, you're welcome to contact Matt and Selena, as Matt just mentioned. And if there are any specific curation questions, they can forward those to me. So there's always help available when using the GCI. So as I mentioned, we'll just walk through each of the different categories of experimental evidence. So the first one, of course, comes under the main category of function and the subcategory biochemical function. So this is evidence showing that the gene product performs a biochemical function shared with other known genes in the disease of interest, or it has a function that's consistent with the phenotype. So one note is that if you are counting this category for biochemical function shared with other known genes, that those other gene products, the function of them must have been proven experimentally and not just predicted. When awarding points in this category, the other known genes should have compelling evidence to support their gene disease association. So if your gene of interest shared a function with another gene which had very, very limited evidence for association with that disease, you might not want to count that. And you can consider increasing points based on the strength of evidence and the number of proteins with the same function that are involved in that same disease. The range of points is 0 to 2, and our default is 0.5. So the first example will be presented by Courtney. So Courtney, do you want to go ahead with this one?
Sure. So this first example here is for biochemical function, and I was looking at the gene PIH1D3, and it was implicated in a disease called primary ciliary dyskinesia. And what we found through this assay is that the PIH1D3 functions in dynein arm assembly. And so disassembly or perturbation of this assembly is known to cause primary ciliary dyskinesia. But more than that, another protein called DNAAF2, which is associated with primary ciliary dyskinesia, has the same function. And so this is where we come into the gene product performs a biochemical function shared with other known genes in the disease of interest, and I gave it default points.
Thanks. So any questions? Okay. All right, so this is another example, and this is an example that I recently curated for the aminoacidopathies group. And actually for this gene, the PTS gene, which is involved in tetrahydrobiopterin deficient hyperphenylalaninemia A, I actually counted evidence under both categories under biochemical function. So on this slide, showing that the gene performs a biochemical function shared with other known genes in the disease of interest. And on the next slide, I'll also show that the gene product is consistent with the observed phenotype. So just to give you some background, the PTS gene encodes the PTPS enzyme, which is involved in tetrahydrobiopterin biosynthesis. So tetrahydrobiopterin is an essential cofactor for phenylalanine hydroxylase, tyrosine hydroxylase, and tryptophan hydroxylase. So without tetrahydrobiopterin, none of these enzymes can function correctly. So individuals with tetrahydrobiopterin deficiency have hyperphenylalaninemia resulting from the PAH deficiency, and they also have dopamine and serotonin depletion in the CNS because the tyrosine hydroxylase and tryptophan hydroxylase are not working appropriately, and this all results in neurological symptoms which are seen in the patients. So the enzyme that is encoded by the PTS gene, which is 6-pyruvyl tetrahydrobiopterin synthase, catalyzes the second reaction in tetrahydrobiopterin biosynthesis. So of interest, this first gene deficiency of the is the GTPCH enzyme, which is encoded by GCH1. Deficiency of this enzyme also causes the same biochemical phenotype and also similar clinical symptoms as well. So because of that, the gene product that I'm interested in, which is the product of PTS, performs a biochemical function shared with other known genes in the disease of interest. I did want to note here that the third step of this pathway, which encodes, synthesizes tetrahydrobiopterin, is performed by the sepiapterin reductase gene. And it's interesting that actually patients with sepiapterin reductase deficiency they don't actually have these features, but what's been shown is that the function of this enzyme can actually be taken over by other enzymes. So that's why these patients don't have similar features to those with the deficiencies in the first two steps of the pathway. Anyway, I felt that this evidence was appropriate for giving default points. As I mentioned, I also counted points for the PTS product deficiency of that being consistent with the observed phenotypes. So as I'd already mentioned, if you have deficiency of this enzyme, of course you can't make tetrahydrobiopterin, and that results in hyperphenylalaninemia because of the PAH not working correctly. You have elevated neopterin because this is the substrate that the enzyme is working on, so this is elevated, and then you have deficiency of tetrahydrobiopterin, and then you have, as I mentioned, deficiency of dopamine and serotonin because you don't have this cofactor for these enzymes, and that's resulting in these neurological symptoms. So all of these biochemical abnormalities are consistent with the function of the PTPS enzyme in the biosynthesis of tetrahydrobiopterin. So I actually felt that, there's a lot of evidence out there. This is a very well-characterized pathway, many papers over the years, so I actually upgraded this to 1 point in this case. I did also want to note that this information actually came from a review, and we have had questions in the past as to whether or not it's okay to curate information from reviews. And I think, the general feeling is that if it's a good review, you're comfortable with the information, and if it's a very sort of well-characterized pathway, well-characterized disease, and this review is really bringing all that information together, that it really is appropriate in that case to curate that information. So that was what I had done here. So any questions about this one? All right, so before we move on, I did just want to show how this looks in the GCI. Again, many of you may be familiar with this, but of course, I had selected here biochemical function, and then it will give you the option to select either A, that the gene product is performing a biochemical function shared with other known genes in the disease of interest, or option B, the gene product is consistent with the observed phenotypes. So for my example, of course, I could do one entry for A and one entry for B and then score appropriately. So moving on, the next subcategory in the function section is protein interaction. So this is where you would be collecting evidence showing that the gene product interacts with proteins that have been previously implicated in the disease of interest. And of course, typical examples of this data could be showing a physical interaction via yeast-2 hybrid or co-immunoprecipitation, et cetera. Of course, the interaction of these gene products must have been proven experimentally and not just predicted. As you may be aware, there are protein interaction databases out there, some of which also include predicted interactions. So we're really only interested in interactions that have been shown experimentally by these experimental methods. And any protein interaction that you're counting, that gene product should have compelling evidence to be associated with the disease of interest. So again, here your range of points is 0 to 2 and default is 0.5. So moving on to some examples, this was an example from Marina. Marina, do you want to go over this one?
Sure. This is just a standard one from the hearing loss group. So this was for the curation CLIC5 and autosomal recessive sensorineural hearing loss. And there is evidence in this paper, the 2014 paper of Salas et al., that CLIC5 interacts with radixin. So radixin is a protein that's definitively associated with sensorineural hearing loss. It actually is a cytoskeletal protein and has been proposed to link actin to the plasma membrane and anchor stereocilia in inner and outer hair cells. So it makes sense that it might potentially be in a complex. So in this case, it was a standard co-IP experiment where both tagged proteins were transfected into, I think, just a standard cell line like COS cells. CLIC5 was express tagged, and TPRN, which is an older name for radixin, was GFP tagged. And as you can see, when you pull down one and the other, they are physically associated together in a complex, at least in this co-IP experiment. And so I had given a standard half a point for this experiment. Something that is maybe a little bit more technical that we hadn't necessarily considered in the hearing loss group, but might be something to consider, is whether endogenous IPs are done or transfected IPs. I know a lot of times technical limitations, you need to do a transfected co-IP with tagged proteins just because of antibodies. So in this case, we weren't concerned about that and just scored it the standard points of 1/2.
All right, thanks, Marina. Any questions or thoughts on that one? Okay. And this is an example from Jen.
Yep. So this is an example for DSC2 and arrhythmogenic right ventricular cardiomyopathy. This paper used GST pull down in rat heart lysates to show that DSC2 interacts with PKP2 and JUP, and both of these have been shown to have a definitive association with ARVC. And I don't have this on here, but it has been repeated in other publications too, so it's pretty convincing evidence. They also showed that a patient variant in DSC2 impairs binding to PKP2 and JUP, which was used towards variant level evidence. But for the interaction with those two proteins, I upgraded this from default to 1 point, since it interacts with multiple proteins that have a definitive association in the same disease.
Great. And Jen, so you put this one in the GCI, right?
Yeah.
And so in the GCI, you can just list all the different proteins that your gene product of interest is interacting with on the same page, right?
Yeah, you can enter multiple genes. So it'll ask in the GCI to type in the gene symbol for the other genes, and then it'll let you type in multiple ones.
Fantastic. All right. Any questions about that one? And since we're mentioning the GCI, of course, this is just what it would look like for the protein interaction section. So just putting your experiment name and then the interacting genes, like Jen had mentioned, interaction type, whether it's the physical, biochemical, genetic, and then methods, and then just outlining more information. So the relationship of the interacting genes and information, where the information can be found in the paper. So again, the GCI is really nice in that it just walks you through all of the information that you need to give. So moving on to expression. So this is where you would summarize evidence showing that the gene is expressed in tissues relevant to the disease of interest, or that the expression is altered in patients who have the disease. And of course, typical methods here are things like looking at RNA transcripts, so RNA-seq, microarrays, qPCR, RT-PCR, etc., and protein expressions such as Western blot, immunohistochemistry. And what we did want to notice here is that expert reviewers may specify appropriate uses of this category depending on their particular disease domain. And Courtney, did you want to make a comment here about, for example, the neurodevelopmental group and how you guys have been approaching this?
Sure. So as Jenny said, it really can be up to the expert reviewer. So in the ID autism subgroup of the neurodevelopmental disorders, larger group, what they've decided is because most every gene can be expressed in the brain, they really wanted to be able to limit that if we counted expression, it had to be isolated to the brain. So unless that particular gene was associated with a greater syndrome. So if, for instance, we found gene A and we had expression in the brain, but kidney and liver, but it was associated with the disease entity of being intellectual disability, we would not actually give points to that unless we could go further, if it was like a northern blot analysis. If we could go further to show that there was Western blot analysis, that it was expressed within neurons in a specific area that we know had phenotypes, we may give partial points. But strictly speaking, for northern blot analysis, RNA, they really wanted it to be restricted to just expression in the brain to get default points.
Great, thanks. So does anybody have any questions about that? All right, so moving on to an example, this is the FAM161A gene in retinitis pigmentosa. And again, I curated a couple of pieces of evidence for expression for this gene. So the expression of FAM161A is expressed in tissues relevant to the disease of interest. Of course, retinitis pigmentosa is a form of retinal degeneration. So this gene actually encodes two protein coding transcript variants, which vary in length. There's a 660 amino acid protein product and a 716 amino acid product. So what had been done in this paper was that they had done a quantitative RT-PCR looking at the two major isoforms. So this is the main one and then the second isoform. And this was done from human tissues. So what they've shown here is that this gene is predominantly expressed in both retina and testis. So it is actually expressed in some other tissues, but at a much lower abundance. And what the authors had mentioned is that testis is an organ that expresses almost 85% of all human genes at various levels, and so their argument was that the biologically relevant FAM161A expression in humans may actually be confined to retina. And they also made note of the fact that there are some other well-known genes involved in retinitis pigmentosa that are not, their expression is not just confined to retina. So because the expression is increased in retina compared to other tissues, I felt that this deserved some points at least, so I gave it default points. There was actually...
Jenny, I have a question on that. Mm-hmm. So are we able to just kind of sort of standardize using like GTEx or one of these large consortia that have done expression and yeah, so that I think I tend to do something like that rather than to look through papers and find it, especially because, I mean, here they only tested a certain, like a handful of tissues versus GTEx is across all of those.
Absolutely, and that's a great question, Deb. And this one I just came across because it was actually this experiment was in the very first paper where they had published patients with variants in this gene. So it was easy; it was in the paper anyway, and they were trying to show evidence that this really is a gene involved in this disorder. But you're absolutely right. I mean, sometimes it can be really, really hard to find good expression studies, or when someone has actually looked at expression across a bunch of different tissues. So yeah, I mean, in the past, I've definitely used NCBI gene, or you could use GTEx, you know, if it's showing you the data that you're trying to find, and then you can just put the PMID for that resource. I've definitely done that in the past.
Well, I didn't know if it was something that, yeah, if we could, like, say, in the same way that we have with exact, you know, define certain levels of, you know, of expression that mean that genes will be expressed or not. So that, I guess I just find using the GTEx resource not as straightforward as I want it to be, first of all, and second of all, I've kind of thought it's a little bit like cheating. I'm like, oh, just running to the GTEx resource, finding it, seeing that, yes, there's some expression I don't exactly know, like the boundaries for what means that it's expressed or not, and I just, like, you know, call that my expression data, give it 0.5 points and move on. I think some better standardization of using that resource would be helpful, and then we can eliminate like searching in deep literature searches, I would say, for expression data from papers, and then just shift to using that resource in the way that we do with exact or something for allele frequency values.
Yeah, I see what you're saying, and you know, I think you know, standardizing it can be tricky because it really just depends on the disease area and what that evidence is. You know, what is the expression of this particular gene, and are your experts comfortable in actually counting that expression data or not? Like, as Courtney was saying, you know, they wouldn't count necessarily just expression in brain because so many genes are expressed in brain. So I think it really is going to depend on the disease area in terms of, you know, what constitutes a threshold beyond which you would say, yes, this is expression, and we would count it.
I agree with that. I agree with that. It's just like in kind of our process, there's nothing that really, I guess, focuses on expression. You know, it just says find expression data and give it a value. It doesn't say consider this resource and make sure that within your field it's this like RPKM value or something like that.
Okay.
Maybe a longer discussion than for right now, but just something I wanted to bring up. And
I think it's a good point. And, you know, also you bringing up GTEx, I mean, maybe that we should try and get someone from that resource on this call to kind of go through it and you know, talk about how it's useful, what the pitfalls are, all of that kind of stuff too.
That'd be great.
Yeah. So let's think about that too. But yeah, absolutely. I hear what you're saying, and it's always nice to fully understand these resources that are available to us, especially for something like expression where you can be kind of rooting through a lot of different papers trying to find something useful. If we have just a go-to resource that's there, that would be helpful. So does anyone else have any comments on that discussion, like using GTEx or NCBI gene or any of these other resources for expression?
So I this actually came up in colon cancer last week. So I had found expression using NCBI gene, and the other curator that was curating the same gene found a paper where they specifically looked in the colon and did not find expression.
Wow.
So I think we need to be careful about using stuff like that, because then we ended up not counting it because it was conflicting, and because the paper, they were specifically looking in the colon. But according to the data on NCBI gene, it looked like there was some expression there, but yeah, we ended up not counting it.
Wow, so that's interesting that there'd be conflicting information. I wonder why that happens. And yeah. Well, thanks for bringing that up, Jen. Has anyone else had similar experience or thoughts on that?
All right. I know this. Oh, sorry, Jenny. This is Courtney. I know just sometimes too. You know, as you were saying, the RPK values just may not be very good either, especially depending on the tissue and can you even get tissue for that. So typically I've used GTEx and NCBI gene as a last resort if I could not find anything in a paper that showed any type of expression, whether it be northern blot or western blot or immunohistochemistry from tissue samples.
Right. So maybe it is nice to have, you know, the two of you are going to use GTEx or NCBI gene, at least, having another source of that information as well if possible. I guess, like we've been saying, sometimes it can be really hard to find good data in papers too, and, how long do you search for before you kind of go to one of these other resources, just trying to balance that. So, all right, well, thanks. That's really helpful discussion. So I did want to mention this other paper as well, so same gene, same disorder, and there were actually a couple of papers that really went on and did a lot of really nice immunolocalization. So as I mentioned, retinitis pigmentosa is a retinal degeneration. So in this particular paper, they had done immunofluorescence studies in both mouse and human and shown that the protein seemed to be localizing between the inner and outer segments of the photoreceptor. And so this is where, thinking about the structure of the photoreceptor, there is what's called the connecting cilium, which basically acts to transport proteins and materials from the inner segment where things are made out to the outer segment, which contains the photoreceptor pigments like the Dopsin. So they're showing that that's where the protein localizes. And then they went on and did additional studies to show that it co-localizes with markers for the connecting cilium, basal body, and the centriole. And then they went on and did some high-resolution immuno-EM, and again showed localization to the connecting cilium. So they had really done a tremendous amount of work in this paper, and there was actually another paper came out around the same time that validated these results. So they got exactly the same information. They used multiple antibodies. You've got mouse and human here. The other paper had actually looked at rat as well. So I had upgraded this to 1 point just because there was so much work there. And with the 0.5 from the other paper, that would give 1.5 per expression for this. So any questions about that one? I guess, one thing that sort of came to my mind, as I mentioned, there was this other paper that had basically shown exactly the same information. They'd done very similar stuff. They came out around the same time. And so, I could curate that one as well. And I could also, give it points as well. So I wondered how people have been sort of dealing with that when you have multiple papers kind of showing the same thing. I mean, you can't just keep, adding points. I guess we have a limit of points anyway, but how have people been dealing with that? I mean, I probably would put both in the GCI and just mention that both are showing similar results, and I may upgrade my points accordingly because it's almost like additional evidence that this really is a true, correct result. But any other thoughts on that?
Yeah, I feel like you can max out expression pretty quickly.
Yeah.
We often try, well, for hearing loss, it's a little difficult because you often don't have human expression unless it's fetal cochlea because it just degrades too quickly when someone's deceased. But often, if we have multiple expression types, maybe we'll try to go across species depending on if it's like a definitive gene curation. We've seen things like mouse and guinea pig and so on. But that we definitely have multiple, I would say, experimental expression points per curation sometimes, but with only the 2, I think it's a 2-point maximum, right?
It is, yeah. So you can just keep, I just keep adding that information until you max out, depending on how you feel that strength of evidence is as a whole. Yeah. Okay.
Sorry, Jenny, I was on mute.
Yeah.
So, I mean, yeah, you can max out expression at 2 points, but then there's like protein interaction and biochemical function too, and the max of all 3 of those is 2. So I just usually get something that would give expression 0.5 points because by then I already have enough with biochemical and protein, and so I guess sometimes I have a question of like, are we curating just to hit the max, or are we curating to like curate the total of literature about this? Many times with the gene, with known genes, I'll never have time to curate all of that literature, and I'm just curating to hit the max.
Yeah, absolutely. And I mean, it's not the goal to curate every single paper. you know, I think it just depends what's out there too. I mean, for some genes, you're just not going to have any protein interaction data. Like if your gene is the only one involved in that disease, it's just not possible to fulfill that category, or there just might not be good biochemical function data. You might not know what it does. So it really, I mean, I think that's the beauty of the matrix. It really does give you that flexibility to decide, you know, what you want to collect, and you can max out that function section just on expression if that's all the data that you have, or you can choose to, if you have that data, you know, score some evidence in the biochemical function and the protein interaction and the expression. So it really just depends what's available to you. But yeah, the goal is not to, you know, curate absolutely everything. I mean, if you've maxed out your points, you don't need to do that. I think one thing that we have tried to do a little bit, especially for the definitive genes, if it's possible, is to try and at least have something in each of those sections. But as I mentioned again, you can't always do that for every gene. It doesn't always exist. So it really just depends on the situation. So any other thoughts on that, on Deb's comment? All right. So this is an example that was from Jen. So Jen, do you want to go ahead with this one?
Yep. So this is for JPH2 and hypertrophic cardiomyopathy. And this is evidence showing that the gene or gene product is altered in expression in patients who have the disease. So this is a Western blot analysis of JPH2 from left ventricular protein lysates. So in the graph on the right, the 3 samples on the left are from our controls. These are from heart samples for people who died of a non-cardiac issue. And then the ones on the right are samples from myectomies from patients that had hypertrophic cardiomyopathy. A few of the samples are from patients that had a MYH7 mutation. Some of them had a MYBPC3 mutation, and then several of them were negative. So across all the samples, though, regardless of the mutation that they had, and then regardless of the gene that the mutation was in, they had a reduced level of JPH2, and they quantified it over all of the patients and showed that this was significant. So, so this was, I gave this a default of 0.5 points, just showing that JPH2 is altered in people with the disease, regardless of what type of mutation they're carrying.
So any questions about that one? Okay, great, thanks, Ben. Again, in the GCI, just a quick screenshot showing that if you choose expression section, you can either score under A, the gene is expressed in tissues relevant to the disease of interest, so that would count for my example with retinitis pigmentosa, and then for B, gene is altered in expression in patients who have the disease, so that would be the one that Jen would have picked. So moving on, functional alteration. So this is evidence showing that the gene and or gene product function is demonstrably altered in cultured patients or non-patient cells carrying candidate variants. So examples of this kind of also include things like gene knockdown, overexpression, etc. And so the scores are slightly different depending on whether the experiment was in patient cells or non-patient cells. So we have an example of each here. So the first one is fibrillin. I think this was your example, Courtney?
Yes, that's correct. So I was curating fibrillin for Marfan syndrome. And part of the phenotypic spectrum of Marfan syndrome is the thoracic aortic aneurysm and dissection. And so this is going to be evidence showing the gene or gene product function is demonstrably altered in cultured patient cells. And what they ended up doing was taking human-induced pluripotent stem cells from Marfan syndrome patients. They had 2 lines in this specific paper that had 2 different fibrillin 1 mutations, and they differentiated these into smooth muscle cells. They then looked at the expression levels of fibrillin 1 and showed that they were the same for them. So they were really trying to figure out what was the mechanism of this mutation. And so we knew that it was expressed the same, but they its ability to actually deposit into the surrounding ECM was changed from wild-type fibrillin versus the fibrillin that came from the Marfan syndrome patient cells. And so that's what you can see here is that in wild-type, we can see the deposition that's occurring, but when we have the patient-derived mutations, there's less deposition. And what's interesting is that the reduced deposition causes extracellular matrix destability for different veins in the aortic cavity. So that's what's leading to the thoracic aortic aneurysm and dissection. And so this one I actually upgraded to the 1.5 point because between the two cell lines showing this, and they also did the immunocytochemistry as well as the ultrastructure to show that there was less deposition in these and that it was disrupted. I felt that it deserved higher points.
Great, thanks. So moving on to Jen's example.
Right, so this is an example in non-patient functional alteration in non-patient cells for LRP6 and non-syndromic oligodontia. And so this is LRP6 is a co-receptor for the Wnt signaling pathway. And the Wnt signaling pathway has previously been implicated in oligodontia. It had been previously found that mutations in Wnt10A cause the same disease. So the pathway has already been implicated. And what they did was express the variant, a patient variant in HEC293 cells and use the top flash luciferase assay to show that this particular variant was unable to activate the Wnt pathway. So since the pathway has been directly linked to the disease previously, I thought this was a good example of functional alteration or evidence showing that the gene or gene product function is altered in non-patient cells.
Well, any questions about functional alteration? Great, thanks, Jen. Oh, and here's another example. This was, Courtney, was this your example?
No, this was mine.
Oh, this is yours. Sorry, Marina. Thanks.
That's okay. I'm impressed you're able to keep them all straight, actually. So this is just another standard one. It's for perilipin-1 and familial partial lipodystrophy. Perilipin-1 is a lipid droplet protein. It localizes to lipid droplets and promotes lipid deposition by inhibiting triglyceride lipolysis. So in this case, 3 patient variants were transfected into the 3T3L1 pre-adipocyte cell line. It's a mouse cell line they used for adipocyte studies. And the cells were loaded with C14 oleic acid. And they found that the cells with patient variants had less lipid droplets, much like the lipodystrophic humans. And also the cells that had the variants in them had less triglyceride when you extract the triglycerides. And also when you measured the release of C14 oleic acid into the media, there was increased release of C14 oleic acid, suggesting that there's increased lipolysis and that this is the mechanism potentially for smaller lipid droplets and potentially for the lipodystrophy. And so in this case, I did give a standard score. However, I could see that because there are multiple variants in this functional alteration that you could upgrade in this case too.
Yep. Cool.
This is Deb. My question is, these are all these are all human cells in some way. So, well, mouse as well. So I have come across yeast expression assays, and I've used that here as non-patient cell data. Is that appropriate, or should we really be looking for human cells, human non-patient cells, or mouse if possible?
I think it probably depends on the disease area. We've had this conversation a lot in the small group in gene curation. I definitely have come across disease areas where it seems like yeast is actually their animal model, essentially, or non-human model organism of choice. So I think it depends on the field. But I do think in some cases yeast is appropriate. But what do others think?
Yeah, I think that's right. And I think we have yeast in the animal model or the non-human model section, right? And I think, yeah, just like you were saying, Marina, it kind of depends on the disease area because, you know, it's hard for a yeast to have a phenotype that would match a human, but if you can find some link to the disease process, it may be appropriate to count it there. We've also used yeast information to count towards the variant or case level evidence as well, so it really just kind of depends on the situation and I guess discussion with the experts.
Well, I'm trying to look in the SOP, like the guideline. Does it say kind of like a hierarchy? Or, I mean, this information that we're saying right now would be really helpful to have in there. Maybe it does. I need to go back and look at it, but
yeah, I don't think we don't have that in there. We can definitely think about adding it. Yeah. Okay, yeah, we can, you know, we periodically make these updates and, you know, this is where it's helpful to get feedback, so we can definitely think about adding some more information in there about yeast. Yeah. All right, any other comments before we move on? So this is what the functional alteration section looks like in the GCI. So of course you have the choice for selecting whether it's a culture patient or non-patient cells, and then based on your selection, it will ask you the additional questions and just walk you through what you need to enter. So moving on, model systems. Of course, this is a non-human model organism, so we often think about things like mouse or xenopus. Our cell culture model with a disrupted copy of the gene that's showing a phenotype consistent with a human disease state. So we've had a lot of questions regarding the difference between cell culture models and functional alterations. So we had added a little bit more guidance in the SOP just to say that cell culture models should recapitulate the features of the disease tissue. So cell culture models could include things like engineered heart tissue or cultured brain slices, as opposed to just, you know, cells that are just growing in a dish. So, and again, we have different range of points depending on the type of model. So if it's a non-human model organism, you can potentially score higher than a cell culture model. So we have some examples here. So this was Marina's example for hearing loss.
Yep, I just wanted to talk you guys through a scenario that we actually come across quite frequently. And if you have any further insight into this scenario, or if you've done something differently, if this happens to you too, feel free to comment. But I just wanted to talk about knockout animal models when the disease is potentially not a loss of function type disease, or variants aren't necessarily loss of function. So oftentimes when we're evaluating animal models, particularly for autosomal dominant sensorineural hearing loss, in this case it's for FLC17A8, it's a solute carrier channel. We often look at the spectrum of variants that are posed to be associated with disease. Oftentimes we'll pull this from HGMD, but you could go to ClinVar, potentially to OMIM, somewhere else to find the variants that are potentially associated in the gene. And so in this case, we had 4 missense variants and 1 small insertion, which does result in a frame shift. And they're all posed to be associated with hearing loss. And another thing we often look at is the constraint scores for the gene when we're thinking about the mechanism of disease from EXAC. At this point, NOMAD doesn't have them, but I know that's coming soon. For hearing loss, it's a little bit difficult because oftentimes constraint scores are best for autosomal dominant conditions. In this case, it is an autosomal dominant condition, but oftentimes the constraint score does take into account reproductive fitness. And in the case of hearing loss, that's not really affected for affected individuals. So we oftentimes have to take the constraint score with a grain of salt. And in this case, the gene is not constrained for missense. Oftentimes, if you think a gene is constrained for missense, a Z-score would be about 3. And something else that we look at is the number of transcripts for the gene, just to keep that in mind when we're looking at variants. And then also the proposed mechanism, like I said. So some authors in this case did propose haploinsufficiency, but as you'll see, the heterozygous knockout mouse doesn't have a phenotype. And most of the variants that are found in humans are missense variants. Next slide, Jenny. So the first mouse model that we found, and this is often typical, was the typical knockout mouse model. So they knocked out the gene using the neomycin cassette for exon 2. And the animals with a knockout of SLC17A8, which is expressed in mouse inner hair cells, were deaf. And this was caused by a lack of glutamate release between the inner hair cells and the auditory nerve. And you can see that there too with the ABR threshold, which is a measure of hearing loss, that the knockouts were up at the top in white, and the hets and wild type individuals were hearing. And so in this case, we actually downgraded the animal model to 1 point because it doesn't necessarily match the proposed molecular mechanism or potentially the variance in humans at this case, in this case. And before we discuss that, I just wanted to talk about the next animal model, which is on the next slide. And so in this case, oh, sorry, this is a rescue. I guess I'll take a side and say that this animal model was rescued by re-addition of the gene, which is also known as VGLUT3. And when they added it back using adeno-associated virus, the mice with the hearing loss.
I'm just going to go to that slide. I think we put it in the rescue section. This is the one that...
Sorry, I wasn't looking at my own slides. My bad.
Okay.
So other evidence that the gene is implicated in hearing loss is when you add it back, you rescue the hearing loss from these animals. Sorry, can we go back to the missense animal model?
Yep.
Thanks.
That was it, right?
So, yes. So in this case, we would prefer actually scoring a missense animal model. And in this case, these authors generated a mouse model with a missense variant in it that was identified in 2 large families with hearing loss. However, in this case, they didn't investigate hearing loss. So all that we could do in this instance, even though we would want to score, I think the authors had mentioned that hearing loss was being investigated, and it was a 2017 paper. So in this case, all we could do was upgrade the points to the variant because they did demonstrate that expression by Western blot in neurons was downregulated with this missense variant. Expression of SLC17A8. So my question is, do others run across this issue, and what have you done? Do you tend to agree that at least the knockout is implicating the gene, is involved potentially in the disease, even if it doesn't recapitulate the mechanism exactly? And would you downgrade to, or have you done other things? Obviously, if we had this missense mouse that had the hearing loss measured, we'd likely score that instead, or maybe further downgrade the other animal model and score this one full points. But we didn't have that option in this case. Maybe in the future we will.
So that's a great example, Marina. So what have other folks been doing in that type of situation? I feel like we've had some examples. Oh, sorry, go ahead.
I was going to say I agree with Marina. I say even the knockout still implicates the gene in the disease, so I just downgrade.
Yeah, this is Courtney. We've done a similar thing in Peds Neuro where we're not exactly having the same mechanism or recapitulation of disease, but if it is showing some kind of cognition impairment, we'll give it partial points like 0.5, so we definitely downgrade it, but it is showing some regulation of that gene with a phenotype associated with the disease.
Okay, that's good to hear. For the rescue, would you guys downgrade that too? I think we ended up giving it standard points because hearing loss was restored in that case, but I could also see downgrading the rescue because we downgraded the animal model itself.
I guess that would seem fine because you're still rescuing, but it's not the full phenotypic spectrum that you're looking for, although it's hearing loss. I think as long as you didn't rescue and then get a whole bunch of other different things, and obviously if you didn't rescue the hearing loss, then I maybe wouldn't count either one of them.
Yeah, that makes sense.
Like you had said.
Great, thanks for that. So this is another animal model. I think
well,
Courtney
that's
was
me.
Yes.
Yes. So what we did was, this is an animal model. It's a mouse animal model. And what it is looking at the gene SCN8A, which is implicated in early infantile epileptic encephalopathy. And so with this specific animal model, it was using the TALEN technology. This was the technology used prior to our CRISPR-Cas9 now, but basically it allows for a knock-in of mutations directly into the mouse locus in the allele. And so the authors here generated mice that were expressing this variant that was associated with EIEE into the mouse. And the mice ended up completely recapitulating the phenotype. They developed seizures and epilepsy very early on, and we can see the changes in their readouts here. And they also, if we look at the table below, and this is with the heterozygous mice, which this is consistent with the diseases being heterozygous, that they ended up getting age of onset of seizures between 8 to 16 weeks, 3 seizures per day, and they ended up having a median age of death at 14 weeks. If you happen to go and delete more than one, you can see that there's an earlier age of death at 3 weeks. So because we now had a patient-derived mutation that was knocked in directly to the locus instead of being overexpressed by a transgene, and we had complete recapitulation of the phenotype, I upgraded this to the maximum of 4 points.
Great, thanks. All right, let's move on. We just have a couple of minutes left. So Courtney, was this is your example too? Yes.
So this might get at what Marina was asking a little bit. Definitely, we were looking into EIEE, and this is another gene KCNT1, which is a potassium channel. And what was interesting about this is that we were looking at mice, and the only mice at this time that were developed for this gene are knockout mice. And what was interesting is that they did show that this gene was required for optimal cognitive flexibility. And so mice in which this gene was knocked out did have some deficits in reversal learning process and ability to adapt. And part of the phenotypic spectrum of the EIEE is not only seizures, but the seizures can lead to intellectual disability. So you would have these learning disabilities. But the thing about it is that the KCNT1 mechanism for EIEE is actually a gain of function of the channel, not a loss of function, and so it doesn't follow the same mechanism. And because this wasn't a phenotype that was very specific to the disease, the experts felt that it would get no points in this particular gene curation expert panel.
Okay, any questions or comments on models? Just have a couple more minutes. So this is a cell culture model. I think this is your example too, Courtney.
Yes, this is mine as well. So in looking at hypertrophic cardiomyopathy, we were looking at FHL1 gene, and what we found is that the authors ended up creating what they called rat-engineered heart tissue. So this is how this will fall under the cell culture model system. So they used rat tissue, engineered it, and then they virally transduced this rat-engineered heart tissue with AAV expressing FHL1 wild type or derived mutations of FHL1. They then went to look at contraction parameters. So they were actually within these tissues looking at how they would contract in beat and the relaxation time. And that can be evident of whether or not there is hypertrophic cardiomyopathy or any kind of cardiomyopathy. And so they found that there were changes to these different contraction parameters. And so multiple variants were tested, and those implicated in both isolated HCM and those associated with Emery-Dreifuss muscular dystrophy, and they saw changes in those parameters. So I ended up upgrading this to 1.5 points because they were showing phenotypes more consistent with the isolated HCM versus the Emery-Dreifuss muscular dystrophy.
Great. Okay, so this is showing the model systems in the GCI. So again, you can select non-human model organism or cell culture model, and then you get appropriate questions after that. So we're actually out of time. We're on the hour now. I know that Marina had already discussed that one rescue experiment. We had a couple of other examples to go through, but what I can do is just put these slides, I guess, on Confluence so people can look at those. We can always come back